Hello everyone, this is Rudree Pathak from Department of Pharmaceutical Technology, LJ Institute of Pharmacy, Ahmedabad. Welcome to the online MOOC lecture series, an initiative taken by LJ Pharmacy, a part of LJ Group of Institutes, Ahmedabad. In today's session, we are going to talk about sterilization of fermentation media. Now, as the name suggests, before we begin with the fermentation process, it is absolutely important to ensure that the fermentation equipment as well as fermentation media is properly sterilized to avoid any kind of contamination of the final fermented product. So let's begin with the session and discuss what are the sources of contamination for fermentation or the chamber or the fermentation media or the fermentation equipment, and how can we actually avoid that, or what are the possible techniques with which we can actually overcome them. Now let's first understand that what are the sources of contamination in the fermentation process. The first source can be fermentation tank itself. That is, if the tank is not properly cleaned or sterilized or made sure that it is free from any kind of disease causing microorganism, it can prove as a source of contamination. Next is if the fermentation media which we are going to use, if it is not properly sterilized before the technique starts, then it can prove to be a very important source of contamination. Then the third and very important is the seals which are present in the fermentation chamber. That is the ports. If you remember the fermentation vessel, there are various entry ports through which actually microorganism can cause contamination. That is temperature control system or the feeding port or the exit port. Now, if we are having n number of ports inside the fermentation chamber, or I would rather say increase in the number of such ports can actually lead to increased chances of the fermentation chamber being contaminated. So let's just try to avoid that. Fourth is improper sterile supply of air. Now, air is an important part if we are dealing with aerobic microorganisms. Okay, we talked about this in penicillin fermentation that the microorganisms which we gonna require are aerobic. So, if we want to ensure a continuous supply of air, we must also have a check that the air should be properly sterilized before use. So, that is also an important source of contamination. And last but not the least is mechanical failure. Now, how that is going to actually interrupt with the overall sterilization process is if we actually have a mechanical failure in between of the sterilization, then we need to stop the process in between, and that leads to entry of n number of microorganisms, which may prove to be a contamination source. So these are the basic sources of contamination which we need to have a check on. Now, let's have an overall look on how these contamination is going to affect the overall fermentation process. Now, as you can see in the chart, the microorganisms or the media or the fermenter itself, as we discussed in the previous slide, can actually lead to contamination. And if it is not checked at the proper time, it may lead to economic loss. The media can get contaminated, which may also result in decreased yield of the fermented product, whether it is any kind of antibiotic or vitamin or any other fermented food product. And that leads to reduced product synthesis, or it may also lead to an altered product, which is not actually important. If you remember in penicillin fermentation, we talked about this that at the time of filtration and separation, any kind of penicillinase producing microorganism should not be there. If at all it is there, it may lead to degradation of beta lactam ring of penicillin, and ultimately it is going to yield a different product which is not actually a penicillin. So we need to have a check on these ports to decrease the disastrous effect of contamination. Now, actually, when and where we can actually have a check on contamination. Now, sterility of media is actually important. So that is the first checkpoint. Then sterility of the incoming and the outgoing air, which we have already talked about this, that the air supply should be properly sterilized before it is utilized in the fermentation chamber. Then sterility of the entire chamber itself is also important because if at all it is rusted or corroded, then it may lead to a different or altered or contaminated product which is not of our use. And overall, during the process, if fermentation is carried out for 15 days or 20 days or any tenure, then during that entire tenure, it is needed that fermentation should be carried out in strict aseptic or strict sterilized environment. Now, what method we are going to use for sterilization? The answer to that is steam. We are going to utilize steam as a sterilization of equipment and media. Now, first of all, you need to keep that thing clear in your mind that sterilization can be carried out by two ways. Firstly, you can have a sterilization of the chamber and media together. Second option is you sterilize the chamber alone and you sterilize the media alone, and then you add your sterilized media to the chamber. That is another alternative. So, whatever option you actually select, steam is the appropriate answer to that. Now, there is a linear relationship between the time of sterilization with the temperature of steam that you are going to take. Higher the temperature, lesser will be the time required for sterilization to carry out. Okay. Now, it is proven that sterilization with steam can actually eliminate all living forms of any transmissible agents of fungi, bacteria, virus, and even spores. So, steam is the proper choice. Now, as we discussed, if we are going for sterilization of production media and steam together, then what would be the conditions? Conditions would be temperature should be kept at 120 degrees Celsius for a time duration of 20 minutes. And the head space. Imagine you are having a chamber in which you have filled your sterilization media. The head space, the space above that fermentation media, should be filled with steam in such a manner that it should avoid any kind of vacuum. The sterilized air which enters inside such a chamber should be entering with positive pressure to avoid any kind of contamination which may result because of vacuum. So these are the points to ponder when we are going for sterilization of steam, sterilization with steam or fermentation media and fermenter together. Now, along with that, we have n number of ancillary equipment parts which are attached to the main fermentation vessel, which needs to be checked upon. First is agitator shaft. That is the shaft with which actually mixing is going to take place in the fermentation media. Then the spargers through which air supply will be given in the media, you need to check on that. Then the defoamer pipelines, that is the pipelines through which defoaming agents, that is the agents which reduces foam in the chamber, actually that needs to be checked on. Then inoculum pipelines, that is the pipeline through which inoculation is going to take place, that is feeding of microorganism. Then the sampling points. Middle of the fermentation process, we need to have some sampling points through which you can actually have a check that okay, fermentation is carried out or it is in the intermediate step or it is already completed, whatever. Okay. And then the unloading ports, which after the fermentation process is completed, you can unload it through that segment. Okay, so these are some of the ports which needs to be checked upon for assurance of sterilization of the entire fermentation process. Next is if we are going for sterilization of fermenter, and production media separately, then it can be done like this. The sterilization of media will be done separately, and it will be based on the composition of media. What ingredients do we have in the media? Based on that, we can select. Say, for example, if the media is having lots of sugar and phosphate, then that needs to be treated separately to avoid any kind of caramelization of sugar. Okay, so these are some of the things which needs to be considered when we are dealing with fermentation media with special requirements. Next is sterilization can be carried out by any of the technique, whether it can be batch or it can be continuous, and it can be done by boiling, by passing off steam, and passing off steam under pressure. Any one you can select, which is given in the PowerPoint presentation. Next is how do we sterilize the air which is going to enter in the fermentation chamber. Now it can be sterilized. The widely used technique for sterilization of air will be always be filtration. Now filtration assures that it is going to restrict the microorganism based on the size of the filter and size of microorganism. Next is you can go for either heat or electrostatic precipitation. I'm sorry for the spelling which is given in the presentation. Please ignore that. And next is UV light based on UV light. And last but not the least, treatment of air with certain amount of chemicals. For more information, do refer videos on sterilization. I think you will be then able to correlate with mechanism of each and every technique which is used. So we are now heading towards the end of the presentation. How do we summarize this? We have discussed what are the sources of contamination in the fermentation chamber, what needs to be treated and what needs to be checked at regular intervals. As you can see, we have a diagrammatic representation of fermentation chamber in the black shown over here. And then we have some ports through which contamination can actually take place. As you can see, we have inoculum feed, then antifoaming agents, then nitrogen, which is our gas or air entry ports. Then we have ejection ports through which waste gases is removed from the fermentation media or fermentation chamber. Then we have sampling ports. Now all of these areas needs to be controlled with sterilization as we have discussed in the entire presentation. Please have this diagrammatic representation clear in your mind to have a quick summary of the entire lecture. Thank you for your patient hearing and stay tuned for more such lectures. Please like and share our YouTube channel Pharma Ignite for more updates. Thank you so much.